Okay, everybody's waiting for me to start the session, I'm just realizing. So welcome everybody. Let's talk about T cells. We are really shifting gears here a little bit and talking about a different modality, and I'm very happy to be sharing this session, also speaking in this session together with very good friends and project partners at the moment. And I think as I'm the first speaker, I leave it to you to introduce me to the audience.
Thank you so much. Yeah, let's open our session with the first talk given by Claudia Rossig. And Claudia is affiliated group leader from Prinses Máxima Center of Pediatric Oncology. And the title of her talk is CAR T cell therapies for pediatric solid cancer. So please welcome Claudia.
Okay, so as I promised, let's talk about T cells and let's dive into the story of CAR T cell therapy for pediatric solid cancers. But to introduce the topic, I'll start a little earlier. As you know, the survival outcomes that we currently see in pediatric solid cancers, which are impressive but with a lot of room for improvement, were essentially obtained with the classical pillars of oncology, anti-proliferative chemotherapy, radiotherapy, and surgery. And molecular targeting has added a little, or is starting to add a little bit of value, but still we need additional options. And T cells, in fact, can act as magic bullets against cancer cells, as we've seen with the checkpoint inhibitors in adult oncology, and may be effectively exploited as an additional option. So since pediatric cancers typically are not infiltrated with endogenous T cells, are not presenting foreign antigens to the immune system. The checkpoint inhibitor story was a failure, essentially, in most pediatric indications. But CAR T cell therapy, where you bring in antigen-specific T cells into the body, could be a feasible and effective alternative. It has taken 30 years from the initial discovery that was made by Zelig Eshar in Israel. He found that a T cell can be tricked into recognizing surface antigens and thereby being activated and lysing the target cell with a recombinant artificial receptor. So that was his observation. Zelig never intended to invent adoptive cellular immunotherapy. He was just trying to find a way to analyze the components of T cell activation. And then this was picked up by clinical researchers like Carl June, but also many others who made additional discoveries that turned CAR T cells into CAR T cell therapy. And the additional discoveries were things like manufacturing the products in a way that you have a safe product to deliver to patients. It was adding co-stimulation to improve the activation response, and also the clinical observation that you need a preparative chemotherapy that depletes regulatory T cells and also has some other effects on the immune system, so that the CAR T cells, once they enter the bloodstream, will find optimal conditions for expansion. And then now, over 10 years ago, in 2013, at the ASH meeting in New Orleans, I still remember that the first cases of pediatric ALL were presented. That sounded like a miracle story. Children with ALL considered incurable, refractory to everything, even potentially transplant, and then relapsing again, emerged with complete remissions, and a lot of these complete remissions were durable in the end. And we all now know patients such as Emily, one of the first children treated, who have survived a disease that was considered incurable at the time. So what is the secret of CAR T cell therapy? I think it's really two features that we should bear in mind. One is that CAR T cells enter the body, meet their target, and then expand initially to high numbers. That's a prerequisite for their ultimate efficacy. And that also means that the dose you apply is not the active dose. The active dose is much higher and is something that happens in the body. So high initial expansion is one feature that is very important. And then these CAR T cells can stay behind in the body as memory T cells and recognize cancer cells again once they emerge in the next month or even years. And that is also something, Emily still has CAR T cells in her blood. She still doesn't have any B cells because her CAR T cells are CD19 specific and deplete B cells. But this is a feature also that is no guarantee for a sustained remission, but something that is associated with long-term responses. So these are the features that we now have to mimic if we want to bring CAR T-cell therapy also to solid tumor patients. Actually, the history, the story of CAR T-cell therapies for solid cancers has started even before the CD19 story. I was a postdoc with Malcolm Brenner at Baylor College of Medicine very long time ago, almost 25 years now. And Malcolm told me to turn an antibody, a neuroblastoma antibody, into a T cell by making a CAR derived from the antibody that we now use for neuroblastoma maintenance therapy. That's the same immunotherapy that the patient story was about in the first plenary session this morning. So we took Ralph Reisfeld's 14G2A antibody and generated a first, still very first generation CAR T cell. And Malcolm lost no time, performed a clinical study, and we just learned that out of this first clinical study, 2 patients have survived long term, even with this first generation CAR T cell product. But overall, the majority of patients failed to respond or had relapses later on. So this was not yet successful therapies, it was just early signs. And then in 2023, so only a year ago, Franco Locatelli and his group published very promising data with an advanced CAR T-cell product that was able to induce complete remission in a substantial proportion of patients with relapsed and refractory neuroblastoma. So all the orange or yellow lines here are patients having complete remissions, and you can tell from the arrows that these were still ongoing at the time of analysis, which is pretty promising and effective. But there is a but even there. It failed to work in patients with bulky disease. CAR T cells were able to shrink bulky tumors, but not to eliminate them. And patients with high burden disease at the time of CAR T cell therapy were not long-term responders. So there is still room for improvement. What can we do about this low performance in CAR T cell therapy for solid cancers? So we have to remember that there are a lot of barriers to overcome. First of all, as I told you, in the ALL setting, we need to achieve this high level of expansion, and the T-cell fitness has to remain for the subsequent months and years. So we have to look at the quality of T-cells to improve it as much as possible. In solid tumors, T-cells have to penetrate into solid cancers, into bulky cancers, and they are not actually homing there because their home is not the solid tumor. The solid tumor has a microenvironment that prevents T cells from entering or inactivates them and finds all kinds of mechanism to protect the tumor from these T cells. So all that has to be overcome, and a lot of research at the moment is going into the understanding of the detailed mechanisms that prevents T cell action in the tumor microenvironment to find ways to overcome these. The approach that we are taking goes back to a German pioneer in CAR T cells, Hinrich Abken. He started designing CAR T cells that, in addition to being activated and removing the tumor cell, also release a cytokine that enhances and broadens their function. He started with IL-12 to activate dendritic cells and enable natural T cell receptor T cell responses. But what we chose to do is engineer the T cells with another cytokine, IL-18, to promote their function, but also activate bystander NK cells, reprogram macrophages. That is the hypothesis, that IL-18 will fulfill all this and thereby overcome more than a single mechanism at the same time. So why IL-18? Well, IL-18 is a very potent danger signal that is employed in the physiological sense by the natural antiviral immune response. And we thought antiviral immune response is really something that is a good template to achieve efficacy against tumors. So IL-18 is synthesized only by myeloid cells and strictly under inflammatory conditions. So when you're first infected, for example, with COVID-19, your body will make myeloid cells produce IL-18, and IL-18 will signal only in T cells and NK cells and will result in higher interferon gamma release, which is also a very potent tool both in virus response, antiviral response, and response against tumors. And then there is an antagonistic mechanism that helps to prevent hyperinflammatory responses. That's a binding protein that abrogates efficacy again because it's so potent. And so our hope is when our CAR T cells release IL-18, it will perform all these functions, strengthen T cells, bystander T cells, NK cells, reprogram macrophages, but then will also be antagonized. And we designed our CAR T cells so that the IL-18 is only released upon engagement of tumor antigen, hopefully in the tumor microenvironment. So we've done preclinical experiments proving some of these points, some of our hypotheses, and this is the key in vivo experiment that we did also to convince regulatory authorities that this is mature for clinical translation. And we found that in a very aggressive neuroblastoma mouse model, where the mice actually don't survive beyond day 10 in the control cohorts, the standard CAR T cells will postpone tumor growth and death, but not cure any mouse, whereas our IL-18 enforced CAR T cells can eliminate the disease, at least under these experimental conditions, and as long as the mice survive under these experimental conditions. So the next step we had to do here to really go to clinic is find a way to manufacture these CAR T cells in an academic setting. And fortunately, there are now automated devices that can be put up in C rooms and hopefully in the future also in D rooms. So you don't really need the infrastructure of classical clean room facilities, which are very expensive to entertain and sometimes even impossible in standard academic labs. So with these tools that can, in a semi-automated way, manufacture your CAR T cell product in a 10 or 12-day process, we were able to set this up in academic settings and obtain manufacturing licenses for our experimental tool here also, GD2 IL-18 CAR-T, in now 2 manufacturing facilities across Germany that provides the trial sites with the product. So the trial sites are distributed all over Germany. This is funded by federal funding, so at the moment it cannot cross borders yet. But of course we are hopeful once this gets to work and fulfills the promise we have, we would go and translate this setting to a European international setting. So it's a clinical phase 1 first in human study that has started in March this year. What we do is we test patients for GD2 expression, patients with either neuroblastoma or osteosarcoma, Ewing sarcoma, and even an adult cohort with breast cancer that can also express high-density GD2. Patients who are GD2 positive, and we've developed a paraffin feasible assay for that, are tested for eligibility and then enrolled. And we have parallel enrollment onto dose levels, the same dose levels, but parallel dose finding cohorts for the pediatric and adult populations. And then we will dose extend for the various entities. That's the design of the trial. And just very briefly, it was initiated at the first site in April. We have 5 sites open. We have 6 patients apheresed, all successfully and also treated. It's way too early to report results, but what I can tell you is that we see no dose-limiting toxicity so far, but some signs of biological activity and inflammatory response. So what can we do about patients who don't fulfill the inclusion criteria by being GD2 negative? GD2 is positive with our assay for eligibility into our study in about 30 to 40% of sarcoma patients, and probably even a little less in breast cancer, and the majority of neuroblastoma, of course, but not all patients will qualify. And also in patients with GD2 positive cancers, probably not all cancer cells in the tumor at all sites will express the target. So antigen negative escape will inevitably occur. So we thought about ways to manipulate also the expression of GD2 in tumors, and we found in Ewing sarcomas that an epigenetic agent, tazemetostat, can inhibit EZH2 and thereby allow re-expression of the enzymes that synthesize GD2, which is not a protein, it's a ganglioside, a carbohydrate essentially. So that can be synthesized again from its precursors when you add tazemetostat. And we showed that with a large number of Ewing sarcoma cell lines, and were able to actually bring up GD2 even in GD2 negative cell lines. And we did this proof of concept in vivo experiments where we injected subcutaneously a Ewing sarcoma cell line that is GD2 negative, then treated the mice with this tazemetostat agent, and then took out the tumor and stained it again and found that it now expresses GD2. And Robbie Meissner in now Boston found the same in neuroblastoma. So there are GD2 negative neuroblastomas, and there will be more when we put pressure on GD2. And he found that tazemetostat can bring up GD2 again also in neuroblastoma. So that is one way of doing this. Overall, with GD2 alone, we will not be able to really make a broad impact in the field because many solid tumors in pediatric oncology do not express GD2. So we keep searching for additional antigens. And my group in Münster has focused on further gangliosides because these are always overlooked in these gene expression assays. People look at gene expression and say we don't find GD2, which is no surprise because it's not a protein. There is no mRNA for it. And so we thought these might be overlooked and looked at embryonal gangliosides and found that SSEA4 is also overexpressed in 2/3 of Ewing sarcoma tissues by biopsies. And when then we looked at the biological features and found that SSEA4 really fulfills all the requirements of an adequate target by being associated with colony formation, with chemo resistance, with migratory properties. So this is just an example that shows it's associated with aggressiveness. And we found that SSEA4 specific CAR T cells are effective to eliminate SSEA4 positive cancer cells in vitro, which is no surprise. Then when we went to the mouse, we found that these cells very effectively kill the mice. And not only eliminate the tumors, and that's what 2 other groups found around the same time. So SSEA4 obviously is not a safe target, not in mice and probably not in humans, but there are now ways to work around this by combining different targets, by designing CAR T cells that are activated by a certain unsafe antigen, but only lyse the cells when an additional antigen is present on the same cell. So this sort of Boolean logic, smarter CAR approaches is something that people are now thinking about, because it is really not very realistic that we will find adequate target antigens surface expressed in tumor cells, but not on any indispensable normal cell beyond GD2 and a couple of others currently investigated. So what if our strategy works, if our GD2 CAR works, if whatever CAR you are designing is working in a phase 1 clinical study? How can we make this, how can we develop this approach to the best of the patients across the world? We've been racking our brains about that as well, because we are realizing that the complexity of manufacturing these products and performing large clinical studies, European clinical studies with an individualized gene and cell therapy product really are tremendous. And we thought that it might be a good idea to enable manufacturing sites in the academic context, the way that we are doing it and have proven the concept that it's feasible across the bubble. Could be Germany at the moment, but the bubble could also be Europe, and the bubble could be the whole world, and establish these centers, and then they will manufacture CAR T-cell products for their own hospital, but not only point of care, but also for other trial sites within their same country, or at least within a distance where the apheresis product can travel and the CAR T-cell product can travel. And if we establish all this across Europe, we will be able to perform even large-scale clinical trials with such novel agents. So that is something we are currently working on. So to conclude, I hope that I could convince you that CAR T-cell therapy does have potential for pediatric cancer therapy beyond ALL. However, advances are definitely needed, and this needs concerted efforts, also in cooperation with industry. We have significant barriers in the tumor microenvironment and need to understand and study these in more details, and that's where molecular targeting and molecular analysis, like in the morning sessions, are coming together with immunotherapy. Antigen negative escape is something that will become a limitation, and we have to work our way around that by looking for better targets, but also making the best of the targets we have. And then last but not least, we have to start thinking now of how we can allow access to effective CAR T-cell products beyond individual countries, and how we can test these products in the academic setting for their best possible use. With that, I'd like to thank my group in Münster and my group at the Maxima, and also the European CAR T-cell Task Force, where I see a couple of members in the audience. So thank you all for listening. I'm happy to take questions, but I think the idea is that I transfer to the next speaker and we answer questions at the end. But if there's something that needs to be clarified right now, I'm happy to answer now.
I would like to thank you for this very comprehensive introduction into CAR T-cell therapies, and also showing the way forward, how these therapies could reach broader patient circles. And any immediate question that we can take now, or we will have a panel discussion at the end of this session. Okay, I think we proceed. Yeah.
Okay, so the next...
Thank you.
So the next speaker is Jochen Buechner from Oslo. Is that right? Even? I don't even have a...
Yeah.
Are you sure?
Yes. Sure. Okay.
Yeah.
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 Yes.
Yeah.
Here
And
you can
so he will. And he will talk about the Norwegian approach to gene-engineered T cells for cancer therapy. Jochen.
Thank you, Claudia, for the introduction, and hello, everybody. I would like to start by thanking the organizers for such a beautiful and important effort to bring all us together. I think this is really a fantastic meeting. Thank you. So this is Oslo. Our hospital is roughly here, and CAR T-cell therapy came to our Nordic area, the NOFO area, in 2015, when Oslo was selected as one of the 7 European study sites to participate in the LIAA trial, which was the Kymriah registration trial. We were from the beginning set up as an international center, meaning that we were able to recruit patients from the whole NOFO area. So we recruited patients not only from Norway, but also from Sweden and Denmark. And at the end, these patients that were all kind of Emily patients, these patients and our Nordic patients were included in the primary analysis that was published in 2018, which was then the basis for the FDA and EMA approval for Kymriah. This is the current label. It then was approved in consecutive in the different Nordic or NOFO countries. Recently, in the context of our NOFO annual meeting, we just collected the data on how we use that commercial product in our NOFO area. So we retrospectively collected data on ALL patients only treated with Kymriah, only with the commercial product, not study patients, and below 18 years, and infused until the end of last year. So we have 5 centers in the Nordics that can give Kymriah. It's Helsinki, Copenhagen, Gothenburg, Stockholm, and Oslo. And I will present only a few of these data on behalf of the centers and the colleagues who collected these data. So very briefly, we we intended to infuse 35 patients. At the end, 34 were infused. And not to go through all the details, but they were 6 years at primary diagnosis and around 9 years at infusion. Some of them were really young, so we had 5 infant ALLs in the cohort. And of course, the majority had so-called typical ALL high-risk features at diagnosis, and a couple of patients were already treated with immunotherapies during primary therapy. So how did they come to CAR T-cell therapy? Well, 4 patients were treated during primary treatment because they had refractory disease. This is a labeled indication. 15 patients received CAR Ts for a first relapse, 7 due to a relapse prior transplant, and then a couple of other patients either due to refractory disease or because the physician decided that the patient wouldn't tolerate a transplant. Some patients had received immunotherapies, and then 14 patients were infused due to a second relapse. Again, some were transplanted, some not, and now they really accumulate prior immunotherapy. They are heavily pretreated, and then 2 patients were infused for a third relapse. And only looking now very not in detail into the outcomes, with a follow-up of almost 2.5 years, 12 patients relapsed post-CAR T. They relapsed both CD19 positive and CD19 negative, mainly CD19 positive, and 13 patients were transplanted post-CAR T, mainly due to relapses. So this would then transfer into an overall survival of 77% and an event-free survival if we take death, non-response, relapse, and if they were transplanted due to loss of the B-cell aplasia as events, our event-free survival in this cohort was 47%. And this is roughly what is reported in the latest follow-up of the LIAA trial, where the 36-month, the probability to remain in remission at 3 years was 52%, and the overall survival at 3 years was roughly 63%. So, well, and some patients had late relapses. So the question is, it works, yes, but patients do relapse still, even after a CD19 CAR and the commercial product. So what are the alternatives? What can we do with these patients, or are there any alternatives to a CD19 CAR? And as it was nicely introduced by Claudia, a CAR recognizes extracellular targets. So it's an artificial receptor that recognizes the target on the outer surface of the cancer cell. And if we use these CARs in therapy, we call it CAR T-cell therapy. By contrast, a natural T-cell receptor is the natural one, not a design, but a natural T-cell receptor works differently. A T-cell receptor recognizes peptides presented on HLA molecules. And these peptides are derived from intracellular or extracellular proteins. So our cells all the time cleave proteins into small pieces and then present these peptides on HLA molecules. And then the T cells with the T cell receptors, they walk around and they recognize these peptides either as something dangerous or not, and will then attack the cell according to the recognized peptide. So T cells, they are able to detect, and this is really, really a huge difference to CARs. A T cell receptor can recognize peptides from an intracellular protein. And the disadvantage is that it is HLA restricted, so it has to be presented on an HLA molecule. So the TCR will always recognize a specific peptide presented on a specific HLA molecule. And if we use TCRs in a therapeutic context, we call this therapy not CAR-T, but TCR, T-cell therapy. And one of these intracellular targets that we believe is a good target for ALL treatment is TDT. TDT is an enzyme that is physiologically used or has its function when the T and the B cell receptor are during the recombination of the T and B cell receptor. So in immature T and B cells, also T cells, it's not expressed in the mature T cells any longer, and it's not expressed in the hematopoietic stem cells. And what is really nice in the context of a tumor treatment is that it's retained in the majority of T-ALL and B-ALL cases. So we use TDT as a marker. We stain for TDT if we evaluate a diagnostic sample. And Johanna Orius, one of our collaborators, she has worked for years on the detection of T-cell receptors that can be used as a therapeutic T-cell receptors. And without becoming too technical, what she has done is she has identified a T-cell receptor that can recognize a peptide from TDT presented on HLA-A2. So this is the identified peptide. So she characterized the peptides presented on HLA molecules, and one of these peptides was peptide 3, and then she isolated a TCR that can recognize this peptide presented on HLA-A2. And this TCR is called T3 in the next slides, just to give you an explanation why there is T3 all the time. And this isolated TCR can then be overexpressed in T cells, and these T cells will recognize the peptide, very, very tiny amounts of this TDT peptide. It's very sensitive. And it's very specific. So these are cell lines. This is a cell line, ALL cell lines, being TDT positive, but HLA-A2 negative. So if they are incubated with T3 T cells, they are not activated. But if you then introduce HLA-A2, they will be activated. And if you knock down the HLA with an antibody, the activation is reduced. And the other way around, if you have a cell line that is TDT positive and HLA-A2 positive, and you knock down HLA-A2, the T cells will no longer be activated. So it's specific for HLA-A2 and specific for this peptide derived from TDT. These are patient samples, 9 BALL and 3 TLL samples from patients, diagnostic samples. And if these samples now are incubated with T3 T cells, you can recognize that the amount of tumor cells, the leukemic cells, they are taken out. So these are the purple bullets here. So they are almost zero in the sample, while the mature T cells and B cells are not affected because they do not express TDT. So they selectively kill the immature leukemic cells. This is again a patient sample number 1, and here all the components in the blood are in one plot, and the patient sample was incubated with the patient's own T cells transduced with this TCR. And you can recognize the red ones are the leukemic cells, and if they are incubated with T3, autologous T3 cells, all the red cells disappear, while the other components are preserved because they do not express TDT. These are mouse models. A mouse that carries two different BALL cell lines, and again, here these mice have leukemia, and over time the leukemia increases the amount of leukemic cells, but if these mice are treated with T3, they are injected with T3 T cells, the leukemia disappears, and these mice survive. And then finally a PDX model, so this is a mouse that carries a patient, ALL, a B-ALL, and again, if these mice are injected with the T3 T cells, at around day 10, the leukemic cells disappear from the blood. And if you then, at the final time point, look into the bone marrow was MRD negative, as well as the blood and the spleen, so there were no leukemic cells left. And how toxic is this? I told you that TDT is expressed in some immature stages during the lymphopoiesis, and these are human thymocytes during their maturation stages from a double negative to a single positive cell. And when they looked into the expression of TDT and HLA-A2, and you can recognize that when the TDT expression is upregulated, HLA-A2 is downregulated at the same time. So there are very few time points, or maybe never a time point when HLA-A2 and TDT, when both are highly expressed. And to further explore this is a mouse who carries a human hematopoiesis. And from this mouse, T cells were collected, and then these T cells were transduced with the TCR, and then they were injected back into the mouse, and then the mouse was sacrificed and looked how does the hematopoiesis is affected of these T cells. And whatever they looked into, the ratio of CD4 and CD8s, the myeloid cells, the B cells, or the colony-forming units, there was no affection of normal hematopoiesis by these T3 cells. So we feel that all has been done in mouse models. Now we should go on and transfer this into a clinical trial. That would be then a phase 1, first in human trial, in which we test these TDT3 cells that we call TDT3 as the final product. This will be a trial that will be initiated in Oslo. It's an investigator-initiated trial. And we will we will offer this treatment to patients who are older than 1 year, who have a relapsed refractory B or T ALL, and we will also include LBL patients. They should not have any other standard treatment options available, of course, because it's a first in human. The cells need to express TDT, and the patient and the cells need to be, of course, HLA-A2 or 1 positive, which is the case in roughly 50% of the European population. They need to have a disease so that we can somehow get first a signal if this treatment gives any efficacy. So in ALL patients, we would require that they have some MRD left, and in lymphoma patients that we can follow the lesion with radiological criteria. They must have recovered from other therapies, and they need to have some T cells, of course, because, and in addition, the first patients need also to have a donor in case that we affect T cells, that these patients can be rescued by a transplant. The product will be, as shown by Claudia, will be manufactured on a prodigy. In T cell, or at least in TDT, TCR, the only effective T cells are CD8 positive T cells, not the CD4 ones. So the apheresis product will be enriched for CD8 T cells, and then these cells will be activated and then transduced and expanded and harvested and then cryopreserved at around day 8, and will then be infused after a standard lymphodepletion. We have a very, very heterogeneous cohort because we have patients that can be 1 year or 73 years. They can have lymphoma, or they can have ALL with 5% or 90% blasts. So we will have a dose escalation scheme, of course, and we will, in addition, give these cells fractionated, meaning that we give 1/3 of the total dose at day 1, and then we give the second 2/3 of the dose earliest on the next day, but we can postpone the infusion for up to 72 hours in case there's very early sign of CRS that does not resolve until the day 3. We will look then in this trial, of course, into the feasibility, if we can manufacture these cells in time for such a high-risk population. And the major focus is, of course, safety in a dose-escalating and fractionated way. And we hope to be able to establish a dose for a phase 2 trial later on. And we aim to enroll 15 patients over 3 years, so not many patients. And we will open the recruitment also for other countries. The TDT manufacturing will at the moment take place in Denmark, in Herlev. And we then plan to transfer the technology after the first patients to our hospital. They have already a valid manufacturing license. So we have a research collaboration with Copenhagen. And patients will receive a bridging therapy until the cells are manufactured and are then infused and followed like in other CAR T-cell trials. So this is my final slide. We hypothesize that TDT is a novel target for a TCR T-cell therapy that can be used both against B and T ALL and LBL. TDT3, the final product, recognizes very specifically TDT that is presented on HLA-A2. We want to move this into clinical development with this trial that is called INSIGHT-1. Currently we are in the phase where we have done validation runs in Copenhagen. So the IMPD will soon be finished. We have had scientific advice meetings, both with the PEI in Germany, because they are very experienced and we wanted to seek advice from them. And we then moved back to our own authorities and got good advice from them in addition. So we hope now to be able to submit the clinical trial application by the end of the year, and we then hope to be able to initiate that trial, let's say, in the second half of next year. I would like to thank many people, of course, the Olveus group who really invented, or Johanna Olveus who invented the whole concept. I would like to thank my colleagues who collected the CAR T data that I presented, our funding bodies, and I would like to thank you for your attention. Paldies par uzmanību.
Yeah, thank you very much, Jochen. Any burning questions now? I actually have one. So this is TDT3. So what was TDT1 and 2? No.
Well, it's only because the peptide was peptide 3.
Oh, okay. In a 96 well plate.
In the Nature Biotechnology, the TCR, there was a TCR1
yeah.
and a TCR3, and
yeah.
TCR3 was more potent.
Okay.
And then we thought we call it TDT3 because it's the T-cell receptor 3 that went on to clinical development.
Thank you for explaining. Anything else? Otherwise, we will move on with Sarah Tasian, who is at the moment also at the Prinses Maxima. Everybody is at the Prinses Maxima at the moment, doing a sabbatical, and who brings in her information, her expertise also from UPenn in the United States, and will talk about not AML as usual, but today about novel strategies for CAR T-cell targeting or immunotherapy of pediatric ALL.
Well, thank you all. Number one, to the organizers, it's a real pleasure to be here in Riga with all of you. It's awesome to focus on precision medicine intensively for a couple of days, but it's also been, I think, a real pleasure for me to learn about the tremendous work that's being done for the children of Latvia in pediatric oncology. I've just been blown away to see your beautiful new hospital building and all of the tremendous philanthropy that's really improving the lives of your patients and families. So thank you. It's a privilege to be here. I am going, if you don't mind, to speak a little bit not just about CAR T cells for ALL, but also just touch on some recent advances in antibody-based immunotherapies that we've appreciated in pediatric B-ALL, simply because I think it is really changing our frontline landscape, and it may also influence the patient populations for whom we are referring for CAR T cells in the future. These are my disclosures, none of which are relevant for today. So just to set the stage, I think, because we have a little bit of a mixed audience, I think that childhood ALL has really been our major success story over the last 50 years of pediatric oncology, and through iterative improvements in clinical trials and understanding how we can put multi-agent chemotherapy together, understanding sanctuary sites and the importance of treating those areas, as well as major advances in risk stratification based upon children's underlying ALL-associated genetics and application of targeted therapies when appropriate. We're really achieving these yellow survival curves now, with upwards of 90% of children being cured in the Western world with frontline therapies. But because B-ALL is the most common malignancy that we see in children, for those who relapse, this is still a substantial patient population, and we know that it remains the leading cause of childhood cancer mortality even in modern times. And I will leave you with a provocative statement, I think, to get started, that we've really enjoyed very remarkable success of some targeted inhibitors and certainly now immunotherapies in children with multiply relapsed B-ALL. And we need to be working together as swiftly as possible to bring these to the front line to the patients who need these therapies if we're to move the needle further. And so when we think about who are the children who need targeted immunotherapies, again, we certainly have learned that there are patient populations with favorable risk genetics who respond very nicely to frontline chemotherapies, and these are patients that we actually know we can reduce their therapy and still cure them with great success, but less toxicity. Some patients need a little bit more intensification of their chemotherapy, but I think it's really these lower two patient populations, those who either have inherent chemotherapy resistance that go on to engender relapse, or for the patients who you might not have predicted a priori, but acquire resistance mutations during the context of therapeutic pressure. These are really the children that have been enriched in our relapse trials of targeted immunotherapies. And I just want to review, just because there's been a lot of excitement about blinatumomab just in the last couple of months for us in pediatric oncology, and I will say in the US it's caught us a little bit by surprise and is changing a lot of our trials, and I wanted to update a little bit about that first before we get into some of the work being done with CAR T cells. So several years ago, based on the success of early phase trials in children with multiply relapsed ALL, we conducted through our Children's Oncology Group, or COG, which is a primarily North American-based childhood cancer organization, a phase 3 clinical trial for children with first relapse of B-ALL. And in this study, which now seems quite boring but was very innovative at the time, we were randomizing children to receipt of standard salvage chemotherapy versus salvage chemotherapy with intercalated blocks of blinatumomab, importantly with removal of some of the very toxic intensive blocks of chemotherapy. And I will be honest that this was initially designed as a non-inferiority trial. We were just trying to understand, could we salvage 60% of patients with blina as we could with chemotherapy? But as we all now know, this was a real home run, and this resulted in about a 15% improvement in event-free survival for these patients, not just because of an improvement in remission rates, many of which were MRD negative, but also because of a reduction in toxic mortality. And this was very exciting for us, and I think really became in North America our new standard of care for first relapse, to use blinatumomab-based therapy. We learned in this study that blina was very effective for our children with medullary relapse or combined relapse, but I think what was very surprising to us is our patients with isolated extramedullary relapse, particularly CNS relapses, did horribly. And I think that this is not completely understood yet. We certainly know that blinatumomab doesn't cross some of those blood-brain or blood-testis barriers, but I think because we reduced the intensity of salvage chemotherapy in this trial, this really resulted in an incredibly poor survival rate with about 30% EFS. PFS, which was worse. So we've abandoned this strategy for patients with isolated extramedullary relapses, and it's gone on to revise some of our current clinical trial strategies right now. But because of the success in the 1331 study, we have gone on to look at blinatumomab in the front line, and we'll come back to that in a little bit. Our colleagues in Europe also at the same time were participating in an industry-supported study that was testing a similar hypothesis that blinatumomab-based therapy for patients with first relapse of BALL could be better. I will note that this was a little bit of a higher risk patient population than our COG one, but the outcomes were the same. Blina was a clear winner, and for patients who received blinatumomab-based therapy, they also had a much higher rate of second remission induction, and in this case, were able successfully to go to transplant. That was an intention of this study. And so these two relapse trials in parallel led to massive FDA and EMA approvals of blinatumomab for relapse indications, specifically in pediatrics, which was very exciting for us. And again, because of the success, and as well as the success of recent data from our adult colleagues who have, unfortunately, I think, jumped in front of us with blinatumomab testing in the front line, I will say that we were quite surprised in June of this year that the US FDA announced an extended approval of blinatumomab, not just in relapse, but for all patients in the front line as consolidation therapy, even as young as 1 month old. And I will say that this was completely based at the time on adult data. We certainly have seen emergent data from the Interfont 06 pilot study, as well as some emerging data that will be presented at ASH from our standard risk 1731 study that I think will convince you that this was a good decision. We also know that CD22 is a really good target for patients with many subtypes of BALL, and at the same time, we've been very interested to understand the therapeutic potential of inotuzumab as an antibody-drug conjugate. And so several years ago, we also conducted a 1621 study looking at the efficacy of inotuzumab given as monotherapy in children with second or greater relapse of BALL. And in the 1621 study, we learned that about 2/3 of children could get into remission with single-agent inotuzumab, which was quite exciting. We also learned that this was a little bit hard on your liver, as you might expect, because of the calicheamycin conjugate. And so I think it just taught us a lot, not just about efficacy of this drug, but also how to manage some of the hepatotoxicity in terms of VOD or SOS risk. But what was very exciting about this drug being safe and tolerable and inducing a high rate of MRD negative remissions is that it has allowed us now to combine inotuzumab with chemotherapy, as well as study it in the front line, and we'll talk more about that in a moment. At the same time, in Europe, our colleagues through the ITCC were conducting a similar trial looking at inotuzumab monotherapy. Their results are actually far better than ours in the COG for some reason, and we don't quite understand why. But in this study, they were able to achieve second remission rates of more than 80%, the majority of which were MRD negative, even with single-agent inotuzumab. Interestingly, they saw almost no VOD and SOS in their study, despite using it at single doses. We still don't quite understand the answer to that. But I think that this has really credentialed inotuzumab as a very important tool in our toolbox for children with relapsed BALL. And based on the success of the ITCC trial, this is also being studied in combination with chemotherapy in successor trials through the Intrial Consortium, as well as in the frontline now through the Altogether Consortium. And based on these data from these two parallel North American and European studies, inotuzumab was given fast track designation and ultimately approved by the US FDA for pediatric relapse indications, and hopefully will receive similar indications by the EMA soon. But of course, we're here to talk about CAR T cells, and so I just want to follow a little bit from Dr. Buechner and talk about some of the successes that we've really seen in BALL. As we all have heard and certainly have appreciated over the last 10 years, I think CD19 CAR T cell immunotherapy has been one of the major successes and really has changed paradigms for us in pediatric oncology, really curing children who had absolutely no chance of survival. And what I put together here is just a summary slide of some of the initial academic clinical trial results from our primarily North American studies, as well as mature data from the LIAA study that Jochen referenced as well. And I think what's really interesting to reflect back now, 10 and 12 years later, is that these were academic CAR T cell products, all using a similar scFv, but very different co-stimulatory domains. And at the end of the day, we saw about an 80 to 90% initial remission induction with all of these phase 1 clinical trials. That was very exciting for us. But unfortunately, unfortunately, only about 50% of patients, no matter which study you were enrolled in, ultimately stayed in remission. And I think what we learned from these phase 1 trials were lessons that we couldn't have predicted from preclinical models and mice. The children really were teaching us important lessons about the mechanisms of immunotherapeutic resistance that we've really realized are quite legion. And so, as you also just heard, there's been very exciting data, I think, especially with the FDA and EMA approvals of Tisigen, Laclusil, or Kymriah. And this has really allowed people to take this to the real world. This is more of a US-based real-world consortium called the PRWCC for short. But I think has taught us now that we all have a little bit better access to Tisigen, Laclusil, how are people really using this in the real world? And are these data that were very carefully controlled in our academic phase 1 trials really translating as we are using this? And what we've seen in these real world data is effectively that the data are the same. We are able to achieve high rates of initial remission induction and then about a 50 to 60% multi-year long-term survival with CD19 relapses, CD19 positive relapses, as well as CD19 negative relapses occurring. But I think what's been really exciting in the real world data is that we now know how to treat patients so much better than we did in those initial phase 1 studies. We are now using CAR T cells with much lower disease burden, which is really mitigating toxicity risk. And I think we have learned a lot more how to treat these patients safely. And so I think this has been a very nice confirmation of those initial, very well-controlled phase 1 data. We've also learned that CD19 CAR T cells, are really not just for multiply relapsed patients. And I think a major push right now is how do we get this to first relapse? And I will say in the U.S. we have been wholly unsuccessful in being able to advance some of these CAR T cells to phase 1, to first relapse clinical trial testing to really be able to access this a little bit sooner for patients, which has been very frustrating. But one place where we have been able to do this is for patients with primary chemotherapy refractory ALL. We all know that children with high-risk BALL who are still end of consolidation or time point 2 MRD positive have very poor outcomes with chemotherapy alone. And as we usually do, for patients who have persistent MRD, we are either giving them blinatumomab and taking them to transplant, but we also have known that this is potentially an opportunity to study CAR T cells in this frontline setting, potentially with a goal of avoiding transplant. As we know, some of the persistent CAR T cell products, such as Tisigen, Laclusil, now are literally lasting for years, and we know that some patients, probably at least 50%, can remain in remission without a consolidative allogeneic stem cell transplant. And so in this international Cassiopeia study that many of our European colleagues have participated in that just completed enrollment, we are trying to test the hypothesis that patients with end of consolidation or time point 2 MRD positivity can be treated with a CD19 CAR T cell without further therapy, and trying to understand for a proportion of patients can they avoid transplant and remain in remission and cure for the long term, as well as what some of the toxicities may be. So I hope that we'll hear outcomes of this trial within the next couple of years. But as I mentioned before, we certainly have learned a lot from the initial phase 1 studies about mechanisms of immunotherapeutic resistance and relapse in these patients after CD19 CAR T cell therapy. And I think what's been really difficult is that there really have been major mechanisms of resistance, and this has not been a one-size-fits-all approach as we try to figure out who these patients are. And at this point, I think all of these factors are really not known a priori before we infuse patients with the T cells. But one of the major mechanisms of resistance that we know is immune rejection. All of the initial CD19 CAR T cell products did have a little bit of murine component of their antibodies, and so immune rejection we know has been a major problem for patients who go on to lose their T cells early or subsequently relapse, usually with CD19 positive relapses. And so one of the approaches or strategies to combat this has been to make those antibodies a little bit more humanized or even fully human. And so several groups throughout the world have conducted now pivotal clinical trials looking at humanized or fully human CD19 CAR T cells to try to overcome some of these issues of immune rejection. And even though these products have not been compared head-to-head in a clinical trial, we have early data emerging that suggests that these humanized or fully human CD19 approaches may actually be superior to Tisigen, Laclusil. Certainly in patients who relapse after Kymriah with CD19 positive disease, we've seen about a 50% success rate of getting those patients back into remission with humanized or fully human products across the trials. But the answers are even better for children who receive a humanized CAR T-cell as their first product, with about an 80% event-free survival. And so we are very curious to understand if this could potentially be a superior strategy, and hopefully some of the trials that are ongoing right now will help us to answer that question, at least in a little bit of an apples to oranges kind of way. And hopefully we'll hear more about this in coming years as well. We also know, as I mentioned before, that CD19 antigen loss is a major problem, particularly with long-term persisting CAR T cells such as Tasian Lechlusel. And the initial emergence of CD19 negative relapse, I think, was very surprising to us. Claudia shared the story of Emily Whitehead, our very first patient treated with Tasian, who went on to have long-term remission and is now a sophomore in college at the University of Pennsylvania, which is very close to home for us. But our second patient on that study was the first patient who developed CD19 negative loss, and I think we don't talk about her quite as much. And she ultimately passed away because we could not get her back into remission. And so a lot of strategies have tried to understand if there are alternative approaches or alternative targets, such as CD22, now knowing the success of inotuzumab also in this patient population. And so many years ago, our colleagues at the NCI developed a first-in-human CD22 CAR T-cell trial to try to understand if this was a viable approach for CAR T-cells as well. And in this study, led by Terry Fry when he was at the NCI, this was really exciting because they achieved a 70% CR rate with a single dose of CD22 CAR T cells. And what I'll note about this trial is that the majority of patients, as you might expect, had CD19 antigen loss. These were incurable patients otherwise. And so this was a really pivotal result for us to see. And these patients largely went on to receive consolidative stem cell transplantation, many of whom were able to survive this. Subsequently, other groups have recapitulated this initial success from the NCI trial with other CD22 academic products and are demonstrating about a 75 or 80% relapse remission induction again, which I think is really nice and gives us, I think, another tool in our toolbox again for these patients. And then if targeting one antigen is good, perhaps targeting two antigens is better. And so a lot of groups have now also developed CD19 by CD22 in the BALL space, also CD20 in some mature lymphoma spaces, co-targeting either bispecific approaches or co-infusion of two monovalent CAR T-cell products. And this was really with the hypothesis that you might be able to mitigate some of that CD19 antigen loss that we've seen with the more persistent products. And I will say that these trials have taught us a lot about the intricacies of cell manufacturing, particularly with some of the bispecific constructs, where the balance of CD19 and CD22 targeting has been a little bit unpredictable, and you never quite know what you get until the products come out. But I think we've learned a lot iteratively through these trials. Unfortunately, I think many of the bispecific trials have demonstrated very similar remission induction rates to that of monovalent CD19 or CD22 CAR T-cell studies. We haven't necessarily seen a higher remission induction rate. And there have been several antigen loss relapses, I would say, in most of the trials until very recently through the CARPAL study led out of London, where so far they have not seen antigen loss relapses with their approach, which I think is going to be interesting to follow. And then the final question, I think, is does your patient need a transplant after CD19 CAR T-cell immunotherapy? And I think that this is probably a philosophical or religious question, depending on your institution. Certainly, many institutions and trials with persistent CAR T-cells are now in the business of trying to avoid the toxicity of transplant for their patients, since we know that about 50% of children in these studies can stay in remission without further therapy, whereas other clinical trials have definitely shown a benefit. And I think it's probably a little bit dependent on the persistence and the stoichiometry of your CAR T-cell product. But certainly there are some very informative data suggesting that some patients really do benefit from a consolidative transplant, which becomes very expensive therapy for the patient. But certainly now, I think even in institutions such as mine, where we are trying to avoid a transplant for many of our patients, there clearly have become some good indications for referring your patient to transplant, such as immune rejection, early loss of B-cell aplasia that we know is a good biomarker for long-term CAR T-cell persistence, concerns for lineage switch that we know are particularly enriched in our patients with KMT2A rearrangements, such as infants, and then with patients, of course, if they relapse and we try to get them back into remission, we generally recommend them for transplant as well. But with that, I hope I've shared with you that I think immunotherapy for us in BALL has been really exciting, and I think we're really moving quite rapidly into the front line, particularly with our antibody-based immunotherapy such as blinatumomab and inotuzumab. And I think it's very curious to understand if we can induce greater remission rates in the front line, perhaps we will have fewer patients who ultimately go on to need some of our cellular therapy options. Again, as I've mentioned throughout this talk, this has been a very exciting time to induce remissions in patients who previously had very few options, but we still very poorly understand some of the subsequent mechanisms of resistance, and I think this is a real opportunity for bedside back to bench correlative studies in a lot of our clinical trials. And then finally, I'll echo some of the points that Claudia made. This is still incredibly expensive and very inaccessible therapies for many patients in need, and I think we really need to take control back within our academic communities to develop more cost-effective and point-of-care mechanisms, and I hope that all of you will help us to solve that. So thanks for your attention.
Yeah, thank you very much, Sarah. Any immediate questions to Sarah? I don't see any, and I think that's good because you made the perfect transition to the next speaker about the complexity of manufacturing the products, and we're all talking about these exciting opportunities, but then it all comes down to manufacturing products and making them accessible. And so Adeel is also from the Princess Maxima. He's heading the cell and gene therapy unit, and he's really hands-on involved in the manufacturing and quality control, and will you give you some insights into that?
Thanks a lot. So yeah, I will also start by thanking the organizers for this wonderful event. And it was really nice to see the new hospital building yesterday. Very impressive progress over the years, so that was great. Okay, so yeah, my talk today, it's a little bit changing from sort of clinical focus to a little bit more non-clinical in terms of actually manufacturing of these products. So I'll give a bit of a whistle-stop tour of what is really needed to set up a facility or a center where you can produce these products and sort of different components of that. Okay, so yeah, I've a few sort of divisions in this presentation. So I'll tell you a little bit about the overall setup, but also from an angle of how we are doing it at Prinses Maxima Centrum. Some novel aspects of our setup. I'll talk a little bit also on systems and equipment that you need, some nuances of cell manufacturing and future outlook at Prinses Maxima Centrum. A bit of a quick personal introduction. So I started my career in blood transfusion back in the UK, and I shifted to stem cell transplantation some 13 years ago. Since then, I worked in 3 different centres in England. Last one was at King's College Hospital in London. And I moved to Prinses Maxima Centrum 2 years ago to set up a new unit for cell and gene manufacturing. So it's a brand new floor. Prinses Maxima Centrum, so it's a centralized hospital that was created by merging 7 other pediatric oncology centres. So they get about 600 new patients every year. And their mission is really to provide a cure for every child, but also maintaining like an optimal quality of life. So they do quite a lot of things in terms of late effects and providing a more holistic approach to treatment. So they had this strategy to really improve on innovative diagnostics and treatment, and there also was immuno-oncology. So that's where this decision was taken to try and invest in cell and gene, and also to have a new center. I'll skip this one. I think we've had a few mentions of Emily Whitehead already. So at Prinses Maxima Centrum, we have about 120 new ALL patients every year. And in terms of AML, there's about 15 to 20. And then a number varies for neuro solid tumor conditions. So in terms of providing these advanced therapy products for these conditions, there's not really a huge interest from large pharmaceutical companies, particularly because these are, particularly in neurosolid tumor, these are rare conditions. So the numbers aren't really there for commercially viable success for pharmaceutical companies. And also looking to sort of recent successes, I think Claudia also shared this, GT2 CAR Ts for neuroblastoma patients in Rome. So really the strategic decision was taken to build our own facility. So this was the ambition for in-house manufacturing, so to manufacture CAR T products. So that would be like our first phase. We have a bit of a pipeline. We also plan to do some graft engineering work for hematopoietic stem cell transplantation, and in future also to expand to some tissue engineering and so products like salivary glands for patients for suffering from late effects after their chemotherapy treatment. So in terms of some of the advantages for in-house manufacturing, so you have a lot more flexibility in terms of when you can schedule a slot, so you're not dependent on manufacturing space in San Diego, and also quite a bit faster in terms of like shaving off some days start and the start of the process, end of the process, because you don't have to ship products over long distances. And also there's some more innovation, flexibility for bringing in more innovation. And in terms of expense as well, so Sarah was talking about like really expensive products. So generally in a academic setting, I think it's getting recognized that you can produce these products at about a third of the price compared to commercial products. So the research building was expanded, and so two new floors were added at Prinses Maxima Centrum, and the sixth floor is for cell and gene therapy. So talking a little bit about, because I know also there's an ambition here to build a manufacturing facility. So there's really two approaches for premises. So you can have like what is called a fully modular approach, where you can sort of like do one type of manufacturing and then move to another room and decontaminate and repurpose it. So there's that, and there's a more traditional way of doing things, which is called stick and build. So we went for stick and build. I think maybe fully modular will be the next one. And not to Baylor College of Medicine. So we actually went with their design for their cell and gene therapy facility. We looked at different designs, and this is the one that we liked the best in terms of flow of materials and personnel and the way areas were segregated. So yeah, this is a floor plan of a bit of a better picture to show you. So we occupy about half of the sixth floor, and this is a simplified layout. So we have 5 clean rooms, 4 grade B clean rooms, 1 C clean room. It's really a purpose-built facility. So we have also our own QC labs, storerooms, cryogenic areas, reception units, and office spaces. And the idea is that everything that we need to manufacture these cells and also test them and release them and so on, we can do all that in-house. So just to talk about these colors, so you have these 4 grades of cleanliness in terms of clean room space. So grade B is basically the cleanest you can have in terms of a room, and grade A are usually like safety cabinets and isolators and so on. Okay, so just to give you an impression of construction, so this was it was a bit of a challenge for them to take all this construction material all the way up to the 6th floor. I think the middle picture there is actually like a window of my office. Some novel aspects. So we have all the different classes in terms of clean room grade. So that's a bit unique and gives us a flexibility. So we can do more high risk open handling steps in grade B, but we can also park a lot of CliniMax prodigies in grade C. So we've got a bit of flexibility. We have a single pass system, so air comes in and goes out, and there's no recirculation of air between clean rooms. So that allows us to be basically a multi-product facility. So we can be doing CAR-Ts in one room, and you could be doing transplant in another room, and we can do doing organoid manufacturing in another room, because essentially there'll be no recirculation. So there's a risk of cross-contamination is nil. And we have also this a system that we developed at King's, and I sort of like brought it with me to Prinses Maxima Centrum, where you can decontaminate rooms using VHP, that's vaporized hydrogen peroxide. So it's about 1,000 times stronger way of disinfecting clean rooms compared to conventional cleaning regimens. Auxiliary areas I already mentioned. We have a mixture of safety cabinets and isolators. Isolators is a bit new way of doing cell processing, so it gives you quite a bit of like assurance in terms of containment, both for protecting staff from aerosols and so on, but also for products. So you essentially have like grade A conditions can be maintained a lot easier compared to safety cabinets. And we thought quite a bit in terms of like flexibility in our design and process flows and so on to essentially future-proof. So current stage is we are, this is what's called a validation model. So you start with your specifications and so on, design, then the actual construction period. Then you have like different qualification steps. So we are kind of up towards the end of our qualification steps. This is what's called PQ, performance qualification. So we're just about to start that. Okay, so moving away from premises construction to talk a little bit about systems. So one of the strengths of, I would say, a dedicated unit that is manufacturing these products is to try and digitalize and automate quite a lot of processes. And that gives you, on one hand, speed. So you could really go from clinical translation to actually viable product very quickly. So we're talking like rather than years, months. And also make all the processes really streamlined, so you don't need like huge teams. So 3 main systems that we are implementing. One is called the Centrum monitoring system. So that's for controlling all our environment in our clean room. So things like temperature, humidity, pressure, gas concentrations. But also implementing an electronic quality management system, because there's really a huge regulatory burden for this sort of work. And then a manufacturing execution system. And I'll quickly show you some slides. So this is a central monitoring system. We have about 180 sensors across the whole facility, monitoring 24/7 all the different parameters. And we have to very tightly control those parameters. This is a bit of a snapshot of our quality management system. So it's things like audits and self-inspections and deviations and so on, and that's all captured electronically. And there's quite a lot of work that goes into to tick all the boxes for regulatory requirements. So we have 2 of my team members, Marlene and Joey, are full-time working on this. So there's it's also a bit like an approach from going from directives all the way to guidelines and so on. So this is one slide showing you what's needed for validating a system. So you have directives, you have like GMP standards, then you have annexes, and then all the way to GAM5, which is a best practice guideline for validating systems for cell and gene therapy manufacturing. In terms of like documents, again, there's a lot of like everything has to be documented in GMP. If it's not documented, it didn't get done. So you have this approach of starting with quality manuals and work instructions and so on, and then at the very bottom you have like all your worksheets and labels and so on. Manufacturing execution system. So here you're tracking all the steps, starting from apheresis all the way up to infusion. And there has to be all these checks built in when you move from one stage to another. So, for example, if you're doing transduction of transduction for CAR Ts, so what is the transduction efficiency and so on. And then once you've met those parameters, then you can move to the next step. Yeah, that's just another slide on that manufacturing execution system. In terms of equipment, so you have integrated equipment and process-related equipment. So integrated are things like safety cabinets, isolators, incubators, and so on. And process-related is things like Clinimax Prodigy. So I'll show you, yeah, this just show a bit of a complexity. So this is an isolator that we designed ourselves for aseptic cell processing, which is all integrated into the fabric of the facility. So that took us like a couple of months. And Clinimax Prodigy, so we bought 2 of these Clinimax Prodigies for CAR T manufacturing. And for one of them, we are doing process validations and so on. So there you are starting with your starting material. So that'd be usually like an apheresis collection. And then you have T-cell enrichment. So in Jochen's group, they're selecting for CD8, but you could be selecting for CD4 and 8 or a mixture of those two. And then, so that's what's called enrichment. And then after that, activating these T-cells. And transduction with viral vectors, usually either lentiviral or retroviral. And then there's an expansion phase, and that's the longest. That's like usually 8 to 10 days. And then after that, you have your final product, and final product needs to be tested for. So usually you have like 10 different tests to do for characterizing these CAR Ts, and are they still sterile in terms of like you haven't contaminated while you've been expanding them for this long. So it's not really like fundamental research as such, but there are some still questions where research questions are kind of like optimization things you can do. So lentiviral transduction is still people are like experimenting what's the best day for transducing, and what's the best like MOI and things like that. So that's often built into process validations. And in terms of manufacturing CAR T is often doses and other things, so like you're actually achieving a viable dose for infusion. And a new aspect is looking into serum-free conditions. So usually you have like human serum, but that's an aspect which adds variability from one product to another. So to actually get rid of this variability, groups are experimenting serum-free conditions. Okay, I think for sake of time, I skipped through some of this because I have some other examples of other type of manufacturing you can do. You can, other than CAR Ts, you can also enrich for antigen-specific T cells. So that's a shorter process, over 12 hours overnight, and so you could, let's say, this is for manufacturing antigen-specific T cells for viral reactivations, for example, post-transplant. So this is one of the things that we may do. And these days, like Miltenyi produces quite a lot of like these GMP-grade peptides. So you incubate your T cells with these peptides, and then they're essentially ready to go after specific viruses. And here's the different conditions you can manufacture these cells for, so things like CMV, adenovirus, ABV, and so on. So that's also something that can be done on Clinimax Prodigy. And this I already mentioned, 12 hours, this Avoscape. I think this is just showing you like the type of enrichment you can have. So you can really enrich for like 80 to 97%. Another thing you can also do is select for certain cells. So, as a boost for transplant, you can select for CD34 positive cells. So this is quite a standard procedure now, and can also be done on Clinimax Prodigy. I'll skip through these. Sorry, I was a bit enthusiastic. I put a lot of slides in. So yeah, this process, cell selection process, is about 4 to 5 hours long. Okay, and last couple of slides. So our organization model. So you really need like these separate teams. So you need like team for QA. This is people who are doing all this quality work. and regulatory work. Then you need people for QC that are testing these products after they've been manufactured, manufacturing team that are hands-on, working in the clean rooms or manufacturing these products. And obviously there has to be quite a bit of interaction with the clinical department as well. So we had quite a bit of extensive planning, and where we are at is that we're aiming for our manufacturing license Q1 of next year, and after that we aim to start with clinical trials. And with that I will finish. And yeah, actually this is one of the trials that we are looking to start with, a PACMAN trial. This is a humanized CD19 for relapsed refractory ALL patients in collaboration with Miltenyi Biotech. And I have some acknowledgements for some of the team at Prinses Maxima. Thank you.
Yeah, thank you, Adeel. I think you really did a very good job illustrating the complexity of it all. Just allow me a provocative question. I'm just coming from a more industrial meeting at Miltenyi Biotech, where a lot of people are considering and listening to us. What we want is more automation, more closed systems. So essentially our dream is to just feed the apheresis product into some black box, and a couple of days later the product comes out and all you have to do is push one button. And do you think that in 10 or 20 years we will even still need such sophisticated clean rooms? And I'm asking this question also because you mentioned that our colleagues in Latvia are thinking about establishing all that. So is this still a good investment? What do you think?
Yeah, it's a really good question. And yeah, I agree. I think the sort of trajectory is that things will be more and more closed system, and so you can come down in these clean room classes, so C and D. I think in our setting, we were thinking it's easier to actually set up for B and then downgrade to C or D than to go other way around, because the whole infrastructure needs to be changed. So I think that was one of the main reasons for having these B rooms, but we can downgrade these B rooms to C rooms in future. And also so for CAR Ts, yes, there's like Clinimax Prodigy and some other machines are also out there. But quite a lot of this work is still quite a bit of like open steps. So transplant work is really like open steps. So if we had ambitions to do transplant work, or let's say this organoid manufacturing, then we need those B rooms. So I think that was the idea that it gives us flexibility.
And the whole all-staff infrastructure is what you definitely need, because it will always be regulated in a very complex manner, obviously. And that is a big part of it. And I think you illustrated that really well, that it's not just one big investment, like spending a lot of maybe donation money on establishing it, and then it's there. It also needs to be run and maintained. But I wouldn't discourage people from doing it, because we, as we sit here, are all believers into the field. Any questions from the audience? I think we don't even have time to sit down and discuss, but please, yeah.
How do you prepare on the IVDR regulatory that will be introduced in 2 to 3 years to the production of relatively personal medicine?
Yeah, good question. So a lot of regulation is changing quickly. So I know there are these new directives that are coming up in the next few years. So yeah, it's basically like trying to future-proof as much as we can. I think we can't predict. Actually, usually the philosophy is to try and see what FDA is doing, because EMA normally follows FDA. So even though actually we are setting it up in the Netherlands, I spend quite a bit of my time looking at these change notifications that I'm signed up to FDA newsletters and so on. So then you see, okay, like what's happening there, and then most likely it's going to happen here in a couple of years' time or 3 years' time. So you have to keep up to date and try to future-proof your facilities, your processes, and so on in preparation for that. Yeah.
Any further questions to any of the speakers?
Hi, I'm Anna Valla and I'm from the local hemato-oncology team. I'm a pediatric hemato-oncologist, and I have a quite practical question. As long as we don't have a local CAR T cell manufacturing site, or what we are actually closer to, we have not had any patient going to CAR T cell treatment anywhere abroad. But we have faced the situation that a very sick patient would be a candidate, but you would need to figure out how to do CAR T cell apheresis. And given that this field is so quickly expanding, given the question that it's not very clear for what amount of population a CAR T facility would be sustainable and so on, would it be, or is it planned that, like for solid tumors, there is autologous apheresis planned for high-risk patients, certain ones that you already, when there is remission, put away when a patient is still in a good condition, do a T-cell apheresis and store it somewhere for places where you don't have CAR T-cell facilities so close by? Or do you think that this is the future a way to do things?
Yeah, do you want to answer? I've been speaking so much already. Or yeah, I think it's, yeah, Jochen, maybe.
I don't know if this is already incorporated into any protocols. We harvest T cells if we have a high risk ALL relapse. We just do the apheresis and put that product into the freezer, because sometimes we see that the patient won't respond, and then we think of CAR T cells, but then all the T cells are gone, and we cannot harvest the patient anymore. There are these washout times, and at the end, the patient will never make it to a CAR T cell therapy. So in some patients, selected patients, we harvest before we start relapse treatment.
Yeah, maybe we should really think of harmonizing this in a way that giving out recommendations. We don't do it in a routine manner, but it has also happened, but we do it not according to a specific algorithm yet, but it's more like someone says, shouldn't we? And so that's not a good way of doing it, but your question was perfectly good to really, it's something that we need to regulate here. Shai.
There was the first question. Sorry, someone before me.
I think there was one before. Yeah, and then you. Okay.
But ask your question first, and then we'll see.
I can ask, well, because this will be a long answer, I'm afraid. What do you think, what does speaker think about allogeneic off-the-shelf, either CAR T or CAR NK cells?
That's more a philosophical question. I personally believe in the full power of T cells with all their features, and when you turn that into an allo product, you will have to do so many things to your T cell that in the end you will have some bullet that has that is devoid of certain characteristics that really make up a T cell. And so you have to deplete them of all kinds of things, make them so that they don't attack the host cells. They are not eradicated by the host cells, and all these manipulations really take you further away from a natural T cell response. So that I believe more in making autologous T cells work. But industry is really trying very hard, because for them it will only become really, from the commercial point of view, a good strategy when you can generate like one product and make it available to hundreds of patients. But I don't have the answer for it. But I think if we really want all the correct characteristics of the T cell with memory and all, then we can't work past that so easily. But I'm happy to hear what my colleagues are thinking about it.
No, I'm Shai. It's a good question. I think, the early experience with allogeneic, mostly CD19 CAR T cells, have been pretty disappointing in terms of persistence. But, for patients, with AML or solid tumors, who are so heavily pretreated where it's difficult or, it's very expensive, to do these, single patient products. I think I agree with Claudia. I think that the technology is so artificial right now that it hasn't fully translated. But if it can be successful, it would be a lot cheaper, faster, and probably better. There are trials going on right now, particularly in T-cell diseases, that, where these manipulations try to mitigate the issues of fratricide. But, one donor is able to treat 10 or 20 patients, and there can be an economy of scale, particularly, if you're in a country where putting all of this, very fancy and expensive technology in just, isn't feasible. So, yeah.
No, but now we are talking about two different types of CAR Ts, right? Third party or allogenic, because
I think this
because
was more off the shelf, the off the shelf question.
yes, because
Make the magic bullet and distribute it to everyone.
right, because the
Sell it in pharmacies.
the other approach to take allogenics, like after a transplant, you go to the donor, is seems to be quite effective, a good approach.
Yeah.
And then there was one more question, the last one.
No?
I think someone here was looking at it. Okay, if no, then
We are past the time, but I think it was a very good session overall. Thank you all for coming. And if you have any questions after that, you can always write emails to either of the speakers.